Hello everyone. Today, I will give a literature review about the Enzyme Engineering Database, simplified as the EnzEngDB. As we all know, the discovery and the engineering of new enzymes is important across the bioeconomy, with diverse applications from foods to pharmaceuticals, sensors to agriculture. However, enzyme engineering, in particular machine learning guided engineering, is hampered by a lack of data. Here, in this paper, the authors developed the Enzyme Engineering Database to provide a centralized resource and an online analysis tool to consolidate sequence function data from enzyme engineering campaigns. In this work, there are three contributions. The first one is that the authors developed such a database into which researchers can deploy public enzyme data. They also provided visualization and analysis tools for protein engineers to analyze their own data or compare enzyme variants to other enzyme campaigns. And certainly, they also provided a gold standard dataset for benchmarking automated extraction along with the first large language model extraction pipeline specific for enzyme engineering campaigns. So they also show that they have provided a gold standard dataset. So for the gold standard dataset, the authors selected peer-reviewed enzyme engineering studies from Arnold's lab from the past decade and have included four additional combinatorial and model-guided mutation datasets that cover diverse enzyme engineering strategies. For the manual curation and cleaning, the curators were three postdoctoral, five PhD, and two master students in Arnold's lab. Curators independently extracted four fields from each paper. The first one is the enzyme variant identifier, and second one is the full-length amino acid sequence. Third one is the model reaction encoded as canonical smells. What are the substrates? What are the products? And what is the name and what is the canonical smells? And they also provided the performance values. There may be some conflicts or discrepancy among different curators, among different papers. So for curators, they sometimes struggle to locate consistent sequence annotations and performance values because the data were scattered across the main text, figures, and supplementary files. Conflicts were resolved by a third curator who consulted the original figures and deployed the consensus record. This manually extracted data ensured consistency between DNA and protein parents and enzyme variants and the validity of the smells using RTK. This is the landing page of the Enzyme Engineering Database. This is a collaboration work between Caltech and Johns Hopkins University. For the Arnold's lab in Caltech, they are mainly responsible for the data curation, for the pipeline construction. For the team in Johns Hopkins University, they are mainly responsible for the web interface development. So for this database, they mainly provided three functions or modules right now. The first one is upload your experiment. The second one is find similar sequences. The third one is explore the database. They provided a web interface about how to upload your experiment and how to upload your data to the platform. For the second module, the authors provided a function called find similar sequences. So given a query sequence, the database would automatically discover the closest matches among all other sequences. As we can see, the first column for the data is the experimental ID, the smells, the experimental name. So this refers to the title of the paper, how much percentage for the match. So this means for the queried enzyme sequence, there are 94.33% match, and they can also provide the substrate smells and the subproduct smells. Furthermore, users can also check the residues with both the gain of function and the loss of function for this detailed mutate residues. And the users can also see the residue with gain of function only or with loss of function only. Apart from this, users can also see the gain of function residue and the loss of function residue in this table. And they can also see the sequence alignment between the query sequence and the matched sequence in the database. And sometimes the residue can also be highlighted in the protein structure here. For the third module in this database, that is explore the database. This means for users, they can know how much dataset they can retrieve in this database. After clicking explore your database, then it would this page would pop out. From this table, we can know how many papers they have connected here. From here, we can know there are around 140 papers that have been connected by manual curation or using large language models. So, for example, when choosing one paper, after clicking the DOI, then users can quickly go to the publication for this paper. For example, for this paper, we can know this is a collaboration paper between Arnold and Hawkes lab. Users can also know more details about the technicals, the data in this paper. After choosing this paper, they can also go to experimental dashboard, and then they can go to the detailed page for this paper. So for this paper, they first need to show the amino acid sequence in this paper, and what is the experimental name, what is DOI, experimental date, uploaded date. Users can also quickly see what is the reaction for this paper, what are the substrate smells, what are the product smells. Furthermore, users can also know the detailed substitution for this enzyme. So, like for this substitution, what is the fitness value 6, 10, and for other mutations, what are the detailed fitness values? Of course, they also provided the wild type enzyme sequence, and then by clicking one substitution, then the detailed mutated site can also be viewed in the protein structure panel. Then this would also be highlighted. And yeah, that's it from my side today. Thank you for listening.